Thank you for your interest in our poster entitled Volumetric Blood Microsampling and LCMSMS Quantitation, a novel approach for the determination of an anti-tuberculosis drug panel, presented at the 2025 MSACL conference in Montreal, Canada. My name is Deema Qasrawi, scientist in method development at Altasciences, and I am pleased to share with you the key findings from our study. Briefly, tuberculosis caused by Mycobacterium tuberculosis remains a major global health threat as one of the leading infectious causes of mortality. Its airborne transmission and increasing drug resistance are major concerns. Standard TB requires long multi-drug therapy, while multi-drug resistant TB require prolonged use of less effective drugs with higher side effects. Bioanalytical assays for TB drugs and their metabolites typically use serum or plasma, which require specialized equipment and infrastructure. Here, we developed a method for the simultaneous measurement of an anti-TB drug panel using dried blood with the B microsampling device. This approach offers volumetric sampling independent of hematocrit, with an engineered inlet to prevent oversampling and pre-cut cellulose discs for easy and reliable sample collection. The objective of our study was to develop a quantitative LCM-SMS method coupled with the B microsampling device to simultaneously quantify 5 anti-TB drugs and 2 metabolites. This physicochemically diverse panel, ranging from highly polar to highly hydrophobic analytes as shown in Figure 1, made both assay development and chromatographic separation highly challenging. The panel includes cetiazolid sulfoxide, cetiazolid, carbodipistat, pretomanid, delamanid, andesmethylbedaquiline, and bedaquiline. During method development for our accelerated stability studies, we stored microsamples at reduced temperature, room temperature, and elevated temperatures under both low and high humidity conditions. We observed that recovery increased as temperature and humidity decreased, suggesting that the changes reflect analyte transformation rather than a true loss in extractability, which is usually linked to alterations in dried blood morphology over time. Literature indicates that delamanid degrades in plasma catalyzed by albumin, and this process can be reduced at pH less than 6. Also, the detection of bedaquiline oxidation products in aged microsamples indicates that the oxidation is a major degradation pathway in these samples. We evaluated different antioxidants and chelators as shown in Table 1. Among them, the 1 molar ascorbic acid provided the highest stability at the highest hematocrit percentage after 6 days of storage at room temperature. For sample preparation, the workflow is illustrated in Figure 2. Briefly, B microsampling devices were pre-treated with 1 molar ascorbic acid. Using its microfluidic design, 25 microliter blood was applied to produce a precise 10 microliter dried blood spot. After overnight drying in a sealed bag with desiccant, the discs were extracted with a solvent containing a mixture of stable labeled internal standards, followed by sonication and centrifugation. The supernatant was then diluted 40-fold and analyzed by the LCMS-MS. During the initial LC development, different LC columns and mobile phases were screened and evaluated. Ultimately, chromatographic separation was achieved using a Phenominex Synergy Polar RP column. The analysis was performed on Shimazu X2 UPLC with gradient elution as shown in Table 2 at a flow rate of 500 microliter per minute with a total runtime of 8 minutes. Overall, the analyte elution order is illustrated in Figure 3, which is correlated with increasing calculated log P for each analyte. The retention time differences enabled the use of MRM in split periods. Detection was carried out using SciEx triple quad 6500 plus, operated in positive ion electrospray mode with multiple reaction monitoring. The approach of period splitting was used to optimize the mass duty cycle, improve sensitivity, and ensure reproducible quantitation. For the method evaluation, we determined the between-run precision and accuracy of the assay at the limit of quantification, as well as at low, medium, and high QC concentrations. The data obtained from 3 independent batches are presented in Table 4 and met all acceptance criteria. For the matrix effect, we evaluated 8 individual donors, including samples with 25% and 55% hematocrit at low and high QC levels. These results shown in Table 5 also met the acceptance criteria. Finally, this novel substrate pretreatment provided 21 days of stability for all analytes by maintaining antioxidant protection and a reduced pH with oxidative products of N-dysmethylbedaquiline and bedaquiline notably absent as shown in Table 6. For our data analysis, we use quadratic regression for the SUS, SZD, QBS, and linear regression for PA, DLM, NDB, and BDQ, as you can see in Figure 4, with an R-square of more than 0.998. In conclusion, this work represents the first application of volumetric microsampling for an anti-TB drug panel. We introduced a novel substrate pretreatment to stabilize labile analytes and employed a straightforward desorption extraction that simplifies the assay workflow. The multiplexed LCM-RMM assay successfully supports a broad range of analytes, and all method evaluation criteria met regulated bioanalytical requirements. Thank you very much for your attention. I'm happy to answer any questions or discuss potential future applications or collaborations. Thank you.